In this module, we will be looking at digestion. Digestion is the breaking down or hydrolysis of large molecules into smaller soluble molecules that can pass across cell membranes and therefore across cells. These molecules can then be absorbed and assimilated into the cells or tissues of the organism. Most digestion of large molecules to smaller molecules takes place outside of cells, so it is called extracellular digestion. Let's look at this process occurring in a saprotrophic fungus. Saprotrophic fungi can be grown on starch agar plates. The agar jelly contains the nutrients needed by the fungus, one of these being starch. We can show that starch is present in one of these plates by flooding the plate with iodine potassium iodide solution. The resulting black color indicates that starch is present. Using aseptic technique, the plate is inoculated with a small piece of fungal mycelium, and the hyphae will grow. If we now flood this plate with iodine potassium iodide solution, you can see an area around the fungal mycelium where the iodine has remained yellow-brown, showing that starch is no longer present. The fungus has secreted carbohydrase enzymes into the starch agar jelly. These enzymes have digested that starch to maltose, and maltose to glucose. Glucose molecules are able to pass into the fungal cells to be used as a source of energy and a source of carbon. Most digestion of food molecules in humans is also extracellular. Cells in the gut wall secrete enzymes into the lumen of the gut, and here many of the food molecules are digested. You may not have thought about this before, but when food is in the lumen of the gut, it is still outside of the body. This may sound odd, but consider the body with the gut running through it. The gut wall is part of the body, but the space or lumen of the gut is continuous with the outside. Food enters this space through the mouth, and undigested food remains, as well as bacteria and dead cells from the gut wall, pass out through the anus. Carbohydrate and protein molecules have to be digested so that the soluble product molecules can pass through the membranes of the cells making up the gut wall and then pass into the blood vessels or in the villi of the small intestine. Fats are digested so that the products of their digestion can pass into the lacteals of the small intestine. Let's have a look at the gross anatomy of the gut. Digestion begins in the mouth. Food is chewed and broken into smaller pieces. This is mechanical digestion and provides a larger surface area for enzyme action. Amylase, secreted by the salivary glands, begins the chemical digestion of starch to maltose. The esophagus is a muscular tube that carries food from the mouth to the stomach. It has no villi or microvilli and no secretory glands. The wall is made of circular muscle and longitudinal muscle. The circular muscles just behind the bolus of the food contract, while those in front of the bolus relax, and this pushes the food along. Bear in mind that in most animals, food is moving upwards in the esophagus as the animal feeds. This muscular action of pushing food along in the esophagus and throughout the rest of the gut is called peristalsis. The stomach is a muscular sac with an inner folded layer, the gastric mucosa, which produces gastric juice. The stomach stores and digests food, mostly proteins. The gastric epithelium has mucus-secreting cells, and in the gastric glands are oxyntic cells that secrete hydrochloric acid and chief cells that secrete protease enzymes, such as pepsin. The hydrochloric acid kills bacteria on the food, and also provides the low pH needed for pepsin to work. Pepsin is an endopeptidase, and it breaks peptide bonds in the middle of protein molecules, breaking them down to smaller peptides and producing more ends on which exopeptidases, produced in the small intestine, can work. In the stomach, the enzyme lipase begins to digest fats to fatty acids and glycerol. Small lipid-soluble molecules, such as alcohol, are absorbed from the stomach, as are some drugs, such as aspirin. After about 4 hours, the pyloric sphincter muscle relaxes to allow the partly digested food, called chyme, into the first part of the small intestine, the duodenum. Here, the bulk of digestion is completed. Bile, secreted from the liver and stored in the gallbladder, enters the duodenum and emulsifies fats to provide a greater surface area for lipase enzymes. Hydrogen carbonate ions in the bile help neutralize the acidic chyme and provide the alkaline conditions needed for digestion in the small intestine. There are no enzymes in bile, but in pancreatic juice there is amylase, that digests starch to maltose, endopeptidases, that digest proteins to peptides by breaking peptide bonds in the middle of the protein molecules, exopeptidases, that digest peptides to amino acids, and these break peptide bonds at the ends of the peptide molecules, and lipase, that digests lipids to fatty acids and glycerol. In the ileum, enzymes are produced and remain on the surface of cells covering the villi. The enzymes are maltase, that converts maltose to glucose, sucrase, that converts sucrose to glucose and fructose, lactase, that converts lactose to glucose and galactose, and exopeptidases, that convert peptides to amino acids. Pancreatic amylase enzymes are also adsorbed onto the glycoproteins in the membranes of epithelial cells covering the villi. The ileum is folded, and there are many tiny finger-like projections called villi, which increase the surface area for absorption of the products of digestion. The final products of digestion, fatty acids and glycerol, amino acids and monosaccharides, can all cross the membranes of epithelial cells covering the villi and pass through these cells and into either the blood capillaries or, in the case of fatty acids and glycerol, into the lymphatic capillaries of the villi. Diffusion, facilitated diffusion, and active transport are all involved in the absorption of the products of digestion. Fatty acids and glycerol are able to pass through the lipid bilayer of the membranes of the epithelial cells by simple diffusion. Inside these cells, they enter the smooth endoplasmic reticulum and are converted back to triglycerides that then pass to the Golgi body to be surrounded by a protein coat forming chylomicrons. These then leave the far side of the epithelial cell and enter the lacteals, or lymphatic capillaries. Glucose is absorbed using active transport and facilitated diffusion. ATP is used to provide energy to pump out sodium ions from the epithelial cells into the surrounding tissue fluid, against the concentration gradient. This lowers the concentration of sodium ions inside the cell, so sodium ions pass through channels in the membrane down the concentration gradient into the cells. They drag glucose molecules with them. This is known as co-transport, and the channels are symports. They transport two types of molecules in the same direction. The glucose then diffuses from the epithelial cells into the blood capillaries of the villi. Amino acids are absorbed in the same way, by co-transport with sodium ions. Fat-soluble vitamins, such as A, D, and E, are absorbed by simple diffusion across the lipid bilayers of plasma membranes. Water-soluble vitamins, such as vitamin C, and inorganic ions, such as chloride ions, pass through transport proteins by facilitated diffusion. Water is absorbed by osmosis. The epithelial cells covering the villi have, in their plasma membranes, microvilli. These further increase the surface area for absorption, as there is more area for protein carrier molecules in the membranes of these cells. These cells also have many mitochondria, as active transport is involved in the absorption of some products of digestion, so much ATP will be needed. Right, now it's time for the quiz.